स्ट्रीक प्लेटिंग के बाद जो दूसरा तरीकाकार बायोटेक्नोलॉजी लैब में यूज़ किया जाता है दैट इज़ कॉल्ड एज कल्चर ट्रांसफ़र। कल्चर ट्रांसफ़र में हम लिक्विड ब्रॉथ यूज़ करते हैं बैक्टीरिया को कल्चर करने के लिए, और वी गो टूवर्ड्स द प्यूरिफिकेशन ऑफ द बैक्टीरियल कल्चर। इस प्रोसेस के लिए हमें एक इनोकुलेटिंग लूप की ज़रूरत है that needs to be sterilized, and the flame sterilization can be done as you can see here. The wire of inoculating loop is heated up to the red, and then this is going to be cooled down for 5 to 10 seconds, but again don't wave it in air to get it cooled down faster. In the second step, after making the loop sterilized, take the tube in which you have established the culture yesterday. Remove the cap of the tube, holding it like in tilted position. When you remove the cap, again maintain the sterilized condition. To maintain the sterilized condition, you need to flame the mouth of the test tube containing your culture. Once the mouth of the test tube is heated on the flame, you can use the sterilized inoculating loop to take the sample from the culture. As you can see in this picture, one tube carries the culture, the second tube carries the uninoculated culture medium. So using this inoculum, take the culture from the culture tube, close the culture tube, and shift your inoculum to the uncultured medium. While taking the sample, try not to touch the walls of the test tube, because if you touch the walls of the test tube, the volume of the inoculum is so small that you may not get anything. Flame the tube lip again and carefully replace the tube cover so that your culture don't get any kind of contamination. As you can see here, these tubes and their lips are being flamed and closed by the cover. Take the inoculum and inoculate the uninoculated culture medium so that you can get your culture further towards incubation. Once you have inoculated your uninoculated culture medium in a new test tube, again to maintain the sterilized condition, you need to flame the lips of the test tube before you place the cover on it. After making it sterile by heating on the flame, cover it with the cover, and now this is ready to be incubated at 37 degrees centigrade or 30 degrees centigrade, keeping in view what microorganism you are working with. Keeping in view what microorganism you are working with. In order to ensure that your working was in sterile conditions, you can repeat step number 1 to 9 using only LB broth as an inoculum instead of bacterial culture. That will act as a control, and in this tube you should not get any kind of growth because you have not inoculated any microorganism in the control tube. Next day or after 48 hours, you can make observation and see if there is any growth in the tubes where you transferred the culture of the microorganism.